started? Good this time? Okay. All right, any questions before we go on? All right. The next thing we want to discuss are the immunoglobulins. And primarily what we're going to be concerned with are structure-function relationships. We're going to talk about the function of immunoglobulins, what they are structurally, and then relate structure and function. And then we'll talk about all the different kinds of immunoglobulins and what they do. All right. First of all, just define the immunoglobulins are these glycoproteins produced by plasma cells, the terminal stage in the differentiation of B cells. And they're made in response to the immunogen, and they function as the antibodies. This is what mediates the humoral immune response. The immunoglobulin molecules are the antibodies. The term immunoglobulin comes from the following observation. If you take serum and you separate serum in an electrical field, the albumin component of serum migrates toward the positive electrode. And migrating toward the negative electrode, there are a series of protein peaks. These were all globular-type proteins. These were called the alpha globulins, the alpha peak, the beta peak, and the gamma peak. Now, if you're to ask yourself, okay, where do the antibodies molecules form? So if you take some serum that you've immunized a rabbit with, all right, say, so it's hyperimmunized with some antigen. And now you take that serum and you run it into things. You'll get what you see in the blue, all right? Now, if you take that serum and you mix it with the antigen that you use to immunize the animal with, and without me telling you how you do this, if you would just give me that, it's possible then to remove those antigen-antibody complexes and remove the antibodies from the serum. And then you rerun it because they're asking, well, what part of the serum is going to be depleted? What you see is what you get in the dotted red line. What you will see is that it is the gamma globulin peak that is reduced, so that the bulk of the antibodies actually are located in this fraction of the serum, the gamma globulin fraction. All right? So these were called the immune globulins or immunoglobulins. You'll also see them referred to the term gamma globulin as used a lot. Somebody gets a gamma globulin shot. What they're getting is this fraction of serum. And we're giving them that fraction of serum because we know that fraction of serum contains the bulk of the antibody molecule. So by giving somebody in a gamma globulin shot, what you're giving them is antibodies to you, so that they would have antibodies to protect them against whatever pathogens you have antibodies in that serum. All right, so these were the immune globulins. Now, the way I want to approach this is to first just talk in some generalities. We'll talk about the general function of all the antibody molecules, and then talk about the general basic structure of all immunoglobulins, and then relate structure and function. Then we'll talk some specifics about the specific different kinds of antibodies. So what do antibodies do? The first and primary function of antibodies is simply to bind antigen. Now, that binding of antigen in and of itself can afford protection. It can confer some immunity. And let me give you some examples. Bacteria. When a bacterium, a pathogen, enters the body, one of the first things that it has to do before it can cause disease, oftentimes, is that it has to attach to some surface so that it can colonize the tissue. So there has got to be attachment. And the bacteria have components on their outer surfaces by which they attach to our epithelial cells. Well, if we can produce antibodies to those components of the bacteria that are involved in this attachment, we can block the attachment. So simply having an antibody that binds to that antigen then will block attachment, will block colonization and prevent disease. So they will be protective and confer immunity. So simply by binding antigen, antibodies can result in protection. And the same thing is true with viruses. Viruses have to attach to our cells before they infect them. If you have antibodies against those attachment proteins that are found on the virus, you block the virus from entering the cell, you block disease. All right? So the antibodies, simply by binding, and all antibodies do this. This is their primary function. Now, when we talk about antibody binding, we need to introduce the term valence. And the valence is nothing else but the number of different epitopes, or the number of epitopes that a particular antibody molecule can interact with. And in the case of all naturally occurring immunoglobulins, the valence is always 2. It may be more, but it's always 2. All right? So the minimum valence for an antibody molecule is going to be 2. Each antibody molecule will bind 2 epitopes. All right? Now, sometimes the binding of the antibody to the antigen does not result in protection. If you have an antibody that's directed against some component of the bacterium that's not involved in colonization, that's not necessarily going to protect. But that doesn't mean that the antibody can't afford some protection. But it is a secondary effect, because it carries out one or more effector functions. So there are secondary effector functions of antibodies by which we can get protected or results in some immunity. And we've actually seen some of these things. We just haven't called them that. Fixation of complement by an antibody is one of these effector functions. By having the ability to fix complement, what do? You start by binding C1q, that whole cascade of events that we talked about occurs. It can result in lysis of the bacteria. So that affords you protection. All right? Binding to various cells. We've already talked about macrophages and neutrophils having on their surfaces a receptor called an Fc receptor for an immunoglobulin molecule. So the antibody binding to that receptor, what did it do? It made the cell more able to phagocytose. In the case of the K cells, binding to the killer cell enabled the cell to kill its target cell. So the antibody's one of these effector functions is the ability to bind to other cells and then modulate the activity of these other cells. So the protection is a consequence of these effector functions. So either we have protection as a consequence of the antigen binding capacity, or protection results from one of these secondary effector functions of the immunoglobulins. All right? Now, in most cases, in order for the antibody to have or demonstrate one of these effector functions, it must have first interacted with antigen. So you have to have antigen binding first. And there are going to be a couple exceptions that we're going to talk about. But by and large, that's true. And if you think about it, it makes sense. If our antibody molecules were fixing complement all the time, what's going to happen? Before they encountered antigen, what would happen if their antibodies would be fixing complement? You'd run out of complement. You'd just be constantly using up all your complement, all right? And so they don't. They only fix complement after they've encountered their antigen, all right? So in most cases, that's going to be true. Again, we'll find a couple exceptions to that rule. All right, so that's what antibodies do. They either bind antigen, or they all bind antigen, and then they may or may not then carry out one or more of these effector functions. And when we talk about the individual kinds of antibodies, we'll talk about what effector functions they can actually mediate. All right. So that's what they do. Now let's talk about their structure. Now, elucidating the structure of the immunoglobulins was actually a pretty daunting task when it was accomplished, because it turns out that the immunoglobulins are an extremely heterogeneous population of molecules. And it's very difficult to try to elucidate the structure of a mixture of molecules. All right? So they're very heterogeneous, and it made the elucidation of the structure actually difficult. The big breakthrough came with the discovery and the realization that myeloma proteins were actually immunoglobulins. All right? Now, do you know what a myeloma protein is? Do you know what a myeloma is? Have you had myelomas? Myelomas are plasma cell tumors, okay? So it's a tumor of a plasma cell. And so these plasma cells would be a clone of cells that would become malignant and would proliferate, and they would secrete a protein, and these proteins were called myeloma proteins. Well, it turns out those myeloma proteins were actually immunoglobulin molecules. And then of course, each myeloma secreted a very homogeneous population of, it only secreted one kind of immunoglobulin. So we had a homogeneous preparation, which we could analyze the structure. And by analyzing these myeloma proteins, it became clear that all immunoglobulins were built up from the basic 4-chain structure, which is indicated here. All immunoglobulins in their basic structure had 2 identical heavy chains, which is indicated in the green, and 2 identical light chains, indicated in blue. And the heavy and light just pertains to the size of these chains. So we have 2 identical heavy chains, 2 identical light chains in the basic structure of all immunoglobulins. These chains are held together by a series of disulfide bonds. There are bonds like this, interchain disulfide bonds like these here. And that's what this red is to indicate the disulfide bond, holding the 2 heavy chains together. Or you may have something like this, a disulfide bond holding the heavy and light chain together. So you had the interchain disulfide bonds. You also had disulfide bonds, intrachain disulfide bonds within the one protein chain. So within the heavy chain, you have a disulfide bond here and here and here and here. So you have intrachain and intrachain disulfide bonds holding this 4-chain structure together. Now, obviously, in addition to these covalent bonds, there are also lots of non-covalent interactions between the protein chains to hold them together. But this 4-chain structure is held together by the non-covalent and as well as the covalent bonds. Now, as more and more of these proteins the myeloma proteins were isolated and began to analyze them, people began to sequence the amino acid sequence of the proteins. And it became clear that both the heavy and the light chain had one part of the molecule that was very variable. If you sequence, say, 10 or 20 of these things and compare the sequences, there's a lot of variability in part of the molecule. But the other part of the molecule was relatively constant. I'm not saying they're identical, but they're relatively constant. So in the case of the light chain, you had roughly about half of the molecule was very variable in its sequence when you compared one to another. So it was referred to as the variable region of the light chain. The rest of the molecule was relatively constant in its sequence, or much less variability. And so it was called the constant region. And the same thing was true in the heavy chain. Part of the molecule was very variable in its sequence, but the remainder of the molecule was more constant, so it was called the constant region of the heavy chain. So each one had variable and constant regions. Now, when the X-ray crystallography was done on these proteins, and you looked at the 3-dimensional structure, there was a region that became to be referred to as the hinge region, because this molecule is not flat as it's depicted here. This acts as a hinge, and these arms can actually move around. They can stretch out. They can move out of the plane and move towards you or move back. So there's movement around this region of the molecule. So we have what is referred to as the hinge region. And these were all glycoproteins, so they all have some sort of carbohydrate. Now, I indicated just one carbohydrate here, but in fact, the carbohydrate can be anywhere. It can be on the light chain. It can be on the heavy chains. It's all over, all right? So these are highly glycosylated proteins. All right. Now, again, the X-ray crystallography showed that this protein is not a flat molecule like we've indicated here, but in fact there are globular regions. There's a globular region, then there's separated by a slightly more linear stretch, and another globular region. And each of these globular regions were referred to as a domain. And it turns out that each of these domains has one of these intrachain disulfide bonds. So in the case of the light chain, there were 2 domains. There was one domain that corresponded to the variable region of the light chain, and one domain that corresponded to the constant region of the light chain. So we have a VL domain and a CL domain. In the case of the heavy chain, one domain corresponds to the variable region of the heavy chain, and the constant region of the light chain there were several domains. So this was referred to as CH1, CH2, and CH3. So we have these domains. And some immunoglobulins actually had another domain out here, a CH4 domain. And I said they were oligosaccharides. All right. So and I'm going to see if this is... So this is a computerized depiction of an immunoglobulin. Now, on my computer at home, I can stop this by right-clicking on it. In the past, I've had trouble for some reason on this computer. I'm going to try to stop this. See, it won't stop. See if yours works. I don't know. Mine on my computer in my office and at home, if I right-click on it, it stops. But you can see the domain structure. In this thing, the The light chains are in green, and the heavy chains are in blue. Although they're two shades of blue, they are identical. They just put two shades in. But you can see the domain. See one domain there? That's the variable region of the light chain. Here's the other variable region, or the constant region of the light chain. You can see the domains, heavy chain VL, CH1. It's more difficult to see CH2 and CH3, which are down here. If you try, if you try it on the thing, see if you're in your right click, if it stops. Does yours stop? This one won't do it. So, all right. So you just play around with it, take a look. You can actually see the domain structure of the immunoglobulin molecules. Oh, this here, the red and yellow, that was that's the carbohydrate that's on there. Okay. Now I want to look a little bit more detail at the structure of just the variable region. So now we're going to forget about the constant region for a while and just concern ourselves with the variable region. As more and more of these molecules were sequenced, and then comparisons were made of the sequence, an analysis was done, which essentially did this. They said, let's look at amino acid position 1. How much variability do I see in amino acid position 1 if I compare 20 or 30 of these different variable regions? And then go to position 2. How much variability do I see in position 2? And then 3, and so on, down all the way down the variable region. What you get, if you make then a plot of the degree of variability as related to the amino acid position, so starting from amino acid position 1 up through about 100, which is about the size of the variable region, okay? What you see is there is variability over the entire variable region. If you were to do the same analysis, for example, on the constant region, this would be a flat line. There would be maybe a couple bumps here and there, but it would be essentially flat, all right? So you can see there's variability all over here. But in fact, there are 3 hot spots of variability, and these are referred to as the hypervariable regions. Hypervariable region 1, and 2, and 3. Notice also that the most variable is hypervariable region 3. This will be important when we talk about the genetics of the immunoglobulin, probably on, well, maybe tomorrow, but more than likely, I guess it's going to be on Friday. All right, so we have these 3 hypervariable regions. These hypervariable regions are also referred to as complementarity determining regions. So why are they called complementarity determining regions? This is the picture that you would get if you sequenced variable regions that came from antibodies of different specificities. In other words, antibodies that were directed against different epitopes or different antigens, all right? This is what you would see. If you did the analysis slightly differently, if you did the analysis by taking only consider antibodies that are directed against the same antigen, all right, and do this analysis, what you will find is that, yes, you see this variability out here, but when you come to this region that corresponds to hypervariable region 1, 2, and 3, it's flat line. There's no variability. So what that says is that antibodies with the same specificity have the same hypervariable regions, whereas antibodies with different specificities have different hypervariable regions. So what that, of course, suggests is that that's the hypervariable regions that determine the specificity of the antibody. Whatever hypervariable region you have, that will determine whatever antigen or epitope that's there, that antibody will react with. And if you do this analysis, you find these hypervariable regions on both the heavy and the light chain in the variable region. You'll see the same picture. And what happens when the heavy and light chain come together, the variable regions come together, these hypervariable regions are positioned in such a way that they create a pocket into which the antigen is going to bind. And they determine the specificity. And that's why these are called complementarity determining regions, because these regions determine which antigen or which epitope that particular antibody will bind. And the specificity is then contributed by both the heavy and the light chain, because both heavy and light chains have these hypervariable regions. And again, they come together to create the place where the where the antigen is going to bind. Okay? Now, the regions flanking the hypervariable regions are referred to as framework regions. So this is framework region 1 and 2 and so on. These are the framework regions. Now, I don't want to say a lot about the framework regions, except to say if you do this sequence analysis and only concern yourself with the framework regions, and you start comparing framework regions, what you can see is if you sequence a lot of these, you might say, these two proteins, these two immunoglobulin molecules in the variable region, they look a lot more similar to each other than they do to this one. This one is really different. So I'm going to put this one in one pile, this one or these two in one pile, this in another pile. And you make a bunch of these things. You'll find out that you can divide the immunoglobulin variable regions into a number of different groups, and ultimately some of those groups were subdivided into subgroups. Well, it turns out that each of those groups and subgroups represents a different gene in the germline for the immunoglobulin molecule. So the framework regions are a way of essentially counting the number of different genes that you have. All right? Okay. So that's what all immunoglobulins are structurally. They're all composed of that basic four-chain structure, and in the variable regions you have these three hypervariable regions that confer the specificity on the antibody molecule. Now let's relate structure to the functions that we talked about. Now the studies to relate structure and function, what they did is they employed techniques where they were basically chop the immunoglobulin up into pieces and then ask what do the pieces do, all right? And then you can identify, what functions each piece carries out. So I'm going to go through and take you through some of the fragments of immunoglobulin molecules. I don't expect you to remember which enzyme you use to generate which kind of pieces, all right? That's not important. What is important is that the pieces, the names and properties of the pieces are important. And the reason is that we refer to the immunoglobulin, and we'll talk about this region of the immunoglobulin, and the regions that we talk about actually come from the names of these pieces, all right? So if we take this structure and we digest it with papain, papain cuts the immunoglobulin molecule, as you're looking at it, to the left of this interchain disulfide bond, these two interchain disulfide bonds that are holding the heavy chains together. As a consequence of cutting there, you generate two different kinds of fragments. These fragments here called FAB, this fragment here called FC. Now, if you take these pieces and then ask, well, what functions do they have? What you'll find is that the FAB fragment of the immunoglobulin has the antigen binding capacity, and that's what FAB stands for, fragment with antibody activity, all right? So this has the antigen binding activity. The valence of the Fab fragments, however, is 1. The valence of the original immunoglobulin up here is 2. The valence of the Fab fragment is 1. You have 2 of those fragments, however, all right? But the valence is different. And, of course, they will bind to the same antigen that the whole molecule will, because the variable regions are contained here, and they have the hypervariable regions that determine the specificity, so everything is going to be the same. These will bind to the antigens in the same way as the immunoglobulin molecule, or the same antigens. The specificity will be identical. The other fragment is called FC. The reason it was called FC is that this fragment was very easy to crystallize, and so this became the fragment crystallizable. This was the crystallizable fragment, and so it was called FC. And so when we looked to see what activities this fragment carries out, and in fact, this is where the effector fragment, or the effector functions map to the FC region. And in fact, in some cases, you can even localize a particular function to a particular domain in there. So, for example, antigen binding is conferred by the variable and heavy variable region in the VH and the VL domains, all right? Complement binding is actually mediated by the CH2 domain in the FC region. And this is why I say it's important to learn these fragments. We talk about this part of the molecule as the FC portion of the molecule. We talk about this as the FAB portion of the molecule, okay? So the complement binding is located in the CH2 domain of the FC portion of the molecule, all right? Binding to the FC receptors, like on macrophages and neutrophils. And that, by the way, that's obviously why they're called FC receptors, because they're a receptor for the FC portion of the immunoglobulin molecule, all right? That is, in fact, mediated by both of these domains. Both of these contribute to the binding. Another one of these effector function is placental transfer. Remember we said that we're not born immunologically competent, that we rely on maternal antibodies. So somehow those antibodies from the mother have got to get into the fetus. And they do so because the FC portion of the molecule, in fact, it's the CH3 domain of the molecule that binds to the a receptor on the placental trophoblast and then is transported across the cell from the maternal circulation into the fetal circulation. Now, this is one of the exceptions to the rule, that it does not require prior exposure to or binding of antigen. So any antibodies that the mother has then can bind to that receptor and be transported over, even though they have not encountered antigen. So that's one of the exceptions. So placental transfer is located to this portion of the molecule. Now, there is one other fragment that I just want to talk to you about, and that's the FAB'2 fragment. If you take the molecule and you digest it with pepsin instead of papain, pepsin actually cuts, again, as you're looking at it, to the right of these interchain disulfide bonds holding the heavy chains together. It also cuts a number of places in the FC portion of the molecule, so you end up with a bunch of FC peptides. The main fragment that you get out looks like this. It's called FAB'2. Obviously FAB because it's got antibody binding capacity. The prime is there because it's different from an FAB fragment, obviously, so it's an FAB prime. What's the 2 for? The valence of 2, okay? So this fragment here has exactly the same antigen binding properties as the original molecule, but it cannot carry out any of the effector functions because it lacks the FC portion of the molecule. All right? Now, again, another point I want to make. These fragments, the FAB fragments, the FC fragments, the FAB'2 fragments, these are only things that are produced in the laboratory. When we make immunoglobulins, we do not make these kind of fragments. In other words, you're not going to find FAB fragments floating around in your serum anywhere. Right? We make whole immunoglobulins. We make these things in the laboratory, but they're very useful because they allowed us to do the structure-function relationship, and because nowadays that's how we refer to immunoglobulin molecules, by these various fragments. Okay. So know what the fragments do. Don't worry about the enzymes that generate them. Okay. So I started out by telling you that immunoglobulins were very heterogeneous, and that's why they were difficult to determine the structure. But yet we've talked about, in generalities, we talked about the general functions of all immunoglobulins. We talked about the fact that they all have the same basic structure. But what is the nature of the heterogeneity? All right, that's what we want to address next. What is the nature of the heterogeneity in the immunoglobulins? And so what we'll be talking about are the immunoglobulin classes and subclasses, types and subtypes. If you sequence all these, when this sequencing is going on, again, now let's concentrate on the constant region and just compare sequences in the constant region. Forget the variable region now. And you sequence a bunch of these myeloma proteins and you compare the sequences in the constant region. What you'll see is that some of them are more like than they are to others. Again, so you can begin to divide them. And you can divide the immunoglobulins into 5 different groups based on the sequence of the amino acids in the constant region of the heavy chain. All right, so we'll look at just at the heavy chain. So IgG, and by convention what is done, the heavy chain is given the Greek designation. So IgGs have a gamma heavy chain. In other words, they have a particular sequence in the constant region of their heavy chain that identifies them as being a gamma chain. All right? IgM has got mu chains. They have a different sequence of amino acids in the constant region of their heavy chains. So the heavy chain determines the class. IgA, alpha heavy chains, D, delta, E, epsilon. So we have these 5 classes of immunoglobulins, each in the classes determined by the constant region of the heavy chain. All right? Now, as more and more of these molecules were sequenced, it became clear that some of these classes can be further subdivided into subclasses. So in the case of IgGs, you can divide IgG into 4 subclasses. gamma IgG 1, 2, 3, and 4 with gamma 1 through 4 heavy chains respectively. Again, this is all based on the constant region of the heavy chain. And in the case of IgA, we could divide the IgAs into two subclasses, IgA1 and IgA2, with alpha 1 and alpha 2 heavy chains respectively. Okay? Now, you can do the same thing with the light chains and sequence the constant region of the light chains and make those comparisons. And what you find if you do that, what you'll find with the light chains, that they fall into two types. The word class was used for heavy chains. The word group was used for the variable region, or the framework region of the variable region. So type is used for the light chain. So we have two types of light chain, a kappa light chain and a lambda light chain. All right. Again, it's based on the constant region of the light chain. And just like with the heavy chains, some of these, as more and more got analyzed, it was clear that some of these can be further subdivided into subtypes. And in the case of lambda, there were 4 subtypes of lambda: lambda 1, lambda 2, lambda 3, and lambda 4. Okay? So we have 4 subtypes of lambda. There are no subtypes of kappa. There's only one type, just one kappa. All right. So an immunoglobulin molecule, again, remember we said is made up of heavy chains and light chains. So the way we name immunoglobulins is to tell you what heavy chain is there and what light chain is there. So for example, an IgM kappa obviously has mu heavy chains, kappa light chains. IgA1 lambda 2 has alpha 1 heavy chains, lambda 2 light chains. Now, here's an important point I'm going to stress, and everybody's going to pay attention and listen. And I guarantee you, I've challenged the class every year. I will catch you on this relatively soon. I won't tell you when, but it's going to be relatively soon. All right? Unless you are told specifically which class or subclass of heavy chain and which type or subtype of light chain is present, you are to assume that everything is there and it is present in all possible combinations. So, for example, if I tell you here's a tube of IgG, I've told you there's only gamma chains there. So there's not going to be any mu chains. There's not going to be any delta or any of those. There's only gamma chains. But I haven't told you anything about the subtypes. So you are to assume that is going to be a population of molecules that has gamma 1, gamma 2, gamma 3, and gamma 4. I've also not told you anything about the light chains. And you know that immunoglobulins have light chains. So there's going to be kappa light chains there. There's going to be lambda 1, lambda 2, lambda 3, lambda 4 light chains there. And those light chains are going to combine with all the possible heavy chains in all possible combinations. So you're going to have a gamma 1 kappa, gamma 1 lambda 1, gamma 1 lambda 2, and so on. All possible combinations. Keep that in mind. So if I were to walk up to you and if I gave you a tube and I said, there's human immunoglobulin here, what's in there? Everything is there. All classes and subclasses are there. All types and subtypes are there. And they're there in all possible combinations. All right? I'll get you. You wait. At least I'm going to get some of you. I know that. All right. So that's the basis of the heterogeneity in the immunoglobulin molecule. It's because we have these different classes and subclasses of heavy chains, types and subtypes of light chains that can mix in all possible combinations. You get a very heterogeneous population of molecules. All right. What I want to do now is, we want to take each of these classes of immunoglobulins and talk about, again, a little bit about the structure, because the structure of some of the classes are slightly different than what we've presented so far. So we'll talk a little bit about the structure and then talk about the major biological properties and functions of each of these classes of immunoglobulins. And we'll start with IgG. As far as the structure is concerned, IgG looks pretty much like what we had already shown, the basic 4-chain structure. You'll sometimes see this referred to as a monomer. Don't get confused. It's not saying that there's just a single chain. When we refer to monomers, when we talk about the immunoglobulins, we're talking about it's built up of one of these basic 4-chain structures. All right? So there's only one of them there. All right? There is an older nomenclature that you may see. and it is referred to, this is oftentimes referred to as 7S. I'm not going to use that nomenclature. I'm just telling you about it in case you do come across it. People refer to this as 7S immunoglobulin. It refers to the fact that if you put this in an ultracentrifuge and spin it down real fast, it sediments at a certain rate, which is based on its molecular weight. And it sediments with a rate of 7S, okay? So that's what that old number, again, I'm not going to use that, but just so you know what it is. So structurally, it looks pretty much, it's built up of this monomer. There can be some differences in the subclasses of immunoglobulin. IgG3 has a particularly large hinge region. You don't need to worry yourself with those kind of things. Just keep in mind there may be some slight differences in the structure of the subclasses, but basically it's a monomer. As far as the properties of IgG is concerned, this is the major immunoglobulin that is found in the serum. It is also the major immunoglobulin that's found in the extracellular spaces. And in fact, this is the major immunoglobulin that protects us systemically. So its major job is systemic protection, okay? Because it is present at the highest concentration in both the serum and in the extravascular spaces. This immunoglobulin is the only class of immunoglobulin that crosses the placenta. All right? None of the other classes of immunoglobulin will cross the placenta. Only IgG does. And again, reminder, this is the exception. The crossing the placenta does not require that there be antigen, that it had first reacted with the antigen. It crosses the placenta without even encountering antigen. Now, again, I don't expect you to remember all the specifics, but just keep in mind that all subclasses don't do it equally well. So I don't want you to remember that IgG2 is the one that doesn't come across. Just remember that they're not necessarily all the same efficiency of transfer. But it is the only class of immunoglobulin that crosses the placenta. It also is a complement-fixing antibody. Again, not all subclasses can do that. IgG4 doesn't, but it's a complement-fixing antibody. As a class, we say it's a complement-fixing antibody. There are a variety of cells that have receptors for the Fc region of IgG. Again, we've seen them. The macrophages have Fc receptors for IgG. The neutrophils have Fc receptors for IgG. And they obviously modulate the function of those cells, okay? Again, not all subclasses do it as well. 2 and 4 don't particularly bind to those receptors very well as primarily 1 and 3. And, of course, they result in opsonization. In the case of the K cells, they result in ADCC. Right? So IgG is actually probably the most important of all the immunoglobulins in the sense that it provides us with our systemic immunity. And it is very versatile in the sense that it really carries out all the effective functions. It's a complement-fixing antibody. There are a variety of cells that bind it, that have Fc receptors for it. And it is also can cross the placenta. So it's the most versatile of all the antibody molecules. And this is actually, when you, in immunization programs, what we're really trying to do is get the production, in most cases, get the production of good high levels of IgG antibodies, because that affords us the most protection. Okay, IgM. IgM structurally is a little bit different. IgM is a pentamer. And what we mean by that, it is built up of 5 of this basic 4-chain structure. All right? So it's a pentamer. But within any individual IgM molecule, every one of these heavy chains is identical, and every one of the light chains is identical. All right? So that means that the specificity of every one of these combining sites is identical. So what's the valence of IgM? 10. Theoretically, the valence is 10 because there are 10 combining sites. In practice, sometimes you see that the valence is a little bit less, because if you're thinking, if you have a large antigen, sterically, if it binds here, it may sterically block another antigen coming in. So theoretically, it's 10. All right, the valence of IgM. Again, the older nomenclature, sometimes you'll see it referred to as 19S immunoglobulin. Again, it sediments at a lot faster rate because it's a lot larger in molecular weight. IgM also is one of these classes of immunoglobulins that has an extra domain in its constant region. So if you look at this is the variable region domain, CH1, CH2, CH3, and there's a CH4 domain. So it's a C-mu4 because this is a mu chain. All right? So it's got an extra chain. So it's a little bit longer, and it's also in this pentameric form. Now, when it exists in this pentameric form, there's also another protein associated with it, and that is, this we indicated here, is J chain. And in fact, this J chain is important in getting this thing polymerized into this polymerized pentameric form. In fact, what you'll see is all the classes of immunoglobulin that are polymerized forms or have multiple units will have J chain. J chain is important in that process. Okay, so this is the J chain. Okay. I tell you what, let me stop here because I don't want to get into the functions. We'll pick up here at this point tomorrow and talk about the functions of IgM.